Good evening, good afternoon, good morning, and it's with pleasure that I introduce both SFA speakers today. As I said, we've got two of them, and both are from Sarah De Beers lab at the University of Antwerp. And first up, we have Irina Spacova, and she obtained her PhD at Leuven before taking up a postdoc with Sarah at Antwerp. And Irina's been a great stalwart of the SFA, serving both an executive committee and a past president. And then after that, we'll have Ilke de Boek. And Ilke got her PhD from Sarah's lab before continuing on with her postdoc there. And so without further ado, I'll invite Irina to start us off. Thank you.
Thank you, Connell, for this introduction, and thank you to ISAPP for this opportunity to present our research on topical lactobacilli against respiratory viruses and their clinical application against COVID-19. So in the first part of this presentation, I will talk about the mechanisms of topical lactobacilli against respiratory syncytial virus, or RSV, which is the topic of my postdoc. And then Ilke, who is working mostly on clinical trials, will talk about our study with probiotic throat spray in COVID-19 patients. So I don't have to explain that respiratory viral diseases are a major global health burden. And for example, RSV puts more than 3 million children in a hospital every year. Most respiratory viral diseases proceed through the same mechanisms. So they start with viral binding and entry, and then go to viral replication and infection spread, airway inflammation and pulmonary damage, and viral clearance and disease resolution. More and more data suggests that beneficial bacteria, and specifically lactobacilli, when applied topically, can inhibit these different stages of respiratory viral disease. And they can do this through direct antiviral activity, through promotion of the epithelial barrier, through micromodulation, and through immune effects. So these mechanisms are something that we summarized in a recent review in Trends in Molecular Medicine. And actually, this balanced antiviral immunity activation is very important in diseases such as COVID-19, because you probably know that they're characterized by a disruption, so a disbalanced response between interferon induction and induction of pro-inflammatory cytokines through NF-kappa-B, for example, that can lead to a cytokine storm. So the first thing that we were wondering was whether lactobacilli can actually stimulate antiviral immune pathways. And we analyzed a range of lactobacilli that were isolated from healthy human hosts for the ability to induce interferon regulatory factor pathways in monocytes. And indeed, they could do that, so they can induce interferon expression in a strain-specific manner. We also looked at NF-kappa-B induction, so this is more pro-inflammatory pathway, and we saw that lactobacilli were also capable of doing that, but to a much lesser degree than the typical pathogens from the respiratory tract, which are more Moraxella and Haemophilus. So from this, we hypothesized that when viruses are combined with these pathogens in the respiratory tract, this can actually exacerbate the disbalance of pro-inflammatory cytokines versus interferon induction. While if we combine viruses with beneficial bacteria, this could promote the balanced responses in terms of cytokines and interferons. Next, we also wanted to look at the direct antiviral activity of our lactobacilli against viruses, and this can happen, for example, through trapping of viral particles, through virus disruption and inhibition, and through competition for host binding sites in the airways. So we did this with Professor Dominic Scholz from KU Leuven. And we made a selection of lactobacilli based on their availability, so commercially or in the lab, their previous application in humans, whether they can occur in the respiratory tracts, their interactions with the airway epithelium, and also their safety. And we combined lactobacilli with RSV, so we pre-incubated them, and then we used the virus pre-incubated with lactobacilli to infect human epithelial cells. And what we saw was that RSV, after pre-incubation with lactobacilli, was actually less cytotoxic to human cells. So the viability of the cells were increased. And this was in contrast with the virus that was simply incubated with PBS or E. coli, which did not infect viral cell toxicity. So this was quite positive. This suggested that our lactobacilli had a direct effect against RSV. And naturally, we wanted to confirm these results in vitro, also in vivo model. And we did this with Professor Peter Delpute at the University of Antwerp. We used a mouse model, and we applied the lactobacilli intranasally for 2 weeks in mice as prophylaxis, and then we infected the mice with RSV. And what we saw was that the mice that were pre-treated with lactobacilli actually had less immune infiltration into the airways after infection with RSV compared to mice that were not pre-treated with lactobacilli. And we specifically saw this at the level of lung eosinophil percentages. So in lung tissues, there are less eosinophils if the mice were pre-treated with lactobacilli before RSV infection. So this suggested that lactobacilli could prevent cellular airway inflammation in an RSV mouse model. And all this work was actually conducted just as the COVID-19 pandemic was starting. So naturally, we wondered whether these locally applied beneficial lactobacilli could also help against COVID-19. So with Professor Dominic Scholz, we combined lactobacilli with a human coronavirus 229 to look at the effects on this virus. And we either added together the lactobacilli with the coronaviruses on human cells, or we first pre-incubated the coronaviruses with lactobacilli and then added them to human cells. And in both cases, we saw that the coronaviruses induced a less cytopathic effect and less cytotoxicity in human cells if they were pre-incubated with lactobacilli. So this suggested that our strains also had direct activity against coronaviruses. And this actually allowed us to make a dedicated selection of lactobacilli strains that could potentially work against human coronaviruses through these different mechanisms. And we wanted to implement them as soon as possible in COVID-19 disease, because we know that the sooner we tackle the disease, the less chance there is of severe consequences. So the virologists were convinced, and we moved on to brainstorming with general practitioners. And this was Professor Samuel Koenen and Professor Wernick Verhoeven from the University of Antwerp. And they were very enthusiastic because they also work with COVID-19 patients, and they told us that there is no new treatment for outpatients besides tender love and care. And actually, 61% of COVID-19 trials are currently conducted in the hospital and not on outpatients. So there was really some kind of treatment needed for outpatients. And we thought that the probiotic spray would be very advantageous for this because it could allow a faster route to the market as an OTC or as a food supplement. So now Ilke will tell you more about the formulation of this spray and the COVID-19 trial that we conducted.
Thank you, Irina. So indeed, for the formulation, we discussed two different options. On one hand, a nose spray, and on the other hand, a probiotic throat spray. For the nose spray, during my PhD and also during my postdoc, we did some previous work for patients with chronic rhinosinusitis. But of course, a nose spray, the nose is an important entry site for SARS-CoV-2. But of course, it's a more complex regulation if we wanted to move on with this nose spray. So therefore, we decided to focus on a throat spray because, of course, the throat is also an important entry site for the virus, and there is a direct link with our lower respiratory tract. This throat spray can then be applied as a food supplement, which is a faster route to the market. And then for the formulation, we worked together with a team of Probiotherapy, and they did experimental work on the taste of the spray, but also on the viability and the stability of the bacteria that we added. And then after a lot of brainstorming and discussion with a very enthusiastic team, we started our cell study in the end of February of this year. And we focused on the patients in primary care with mild to moderate symptoms. It was a study that was completely remote self-sampling. So once a participant started in the trial, we drove around and we delivered study packages at their place, and they had all the materials for the entire study in this study package. The trial is a randomized, double-blind and placebo-controlled trial, and we did not have any dedicated funding for this trial. So this is a research-driven trial with thanks to a very enthusiastic team of researchers. And what we want to investigate is whether we can apply these probiotics topically and soon enough to reduce the duration and the severity of COVID-19 symptoms. In addition, we also want to explore whether these topical lactobacilli can reduce the viral loads, whether they can have an impact on the antibody responses, and also whether they can have beneficial effects on our airway microbiome. For the inclusion criteria, it was important that they had a positive PCR test and that they were recruited within 72 hours after their test results. And here you can see the map of Belgium, very tiny. And then we focused on the province of Antwerp, so you can see the location of our campus. And we were driving around, myself, but also a lot of my colleagues, we were driving around to deliver all these study packages to our participants. And in February in Belgium, the COVID numbers were high, but actually the recruitment was still a major challenge. So we made use of different ways to recruit our participants. We had a very dedicated team of general practitioners, but we also went to the local triage centra to promote our study via flyers, and we had a press release of the study, which was really a great success. Then in the end, we assessed 112 individuals for their eligibility, and of these 112, 79 were randomized in the A spray or the B spray group. After the dropout, 65 patients completed the entire trial. And what we asked them to do is to use a probiotic throat spray for 2 weeks, and then in the last week, they did not have to use the spray anymore. But they had to fill in an online diary for 3 weeks where they noted down their symptoms and their severity. And for that, they received an email every day with a link to their personal diary. Like I mentioned before, this study was completely remote and self-sampling. So our participants took at 4 different time points in the study a nose throat swab. And at the beginning and the start of the study, they also collected a blood finger prick. And here are some first results on the study compliance. And as you can see in the first graph, is that our participants actually did a very good job in filling in their online diaries almost every day. Also, the throat spray, we advised them to use it up to 5 times a day, and most of them also did it 4 to 5 times, and if not, at least 1 to 3 times a day. And actually, from last week on, all the samples are back at our lab. So meaning we were driving again around the province of Antwerp and collecting all the samples, and they are now stored in our lab. And also here you can see the amount of samples we received is a good way to show that the study compliance was good. And currently we are doing a lot of data processing. Last week, our samples for RNA extraction and the determination of the viral loads were sent to the Antwerp University Hospital. Next week, in our own lab, we are planning the DNA extractions and the preparations for the sequencing. And also the blood finger prick samples are currently at the Institute of Tropical Medicine. So a lot is going on for this trial, and we are really looking forward to our results. And of course, we are all living now in this pandemic, but I want to end up with this slide to show you that actually there is a lot of potential for probiotics in our airways. And we have been doing quite some research on it for the last years before the COVID pandemic as well. So I did my PhD and I focused my postdoc on chronic rhinosinusitis. Irina has worked on allergic asthma and is now focusing on RSV. But also the colleagues Eileen and Joke are working on airway probiotics. Eileen is focusing on cystic fibrosis and Joke on otitis media. So as you can see, a lot of potential for airway probiotics. And with that, I would like to end, and I would like to thank a lot of people. And of course, I would like to thank you for your attention.
Thank you very much, Irina and Ilke. We seem to have one question. We've just about enough time for it. That's from Gregor Reed. And he is asking, what is the shelf life of the product?
That's a very good question. And the product can be at least stable for, I think, because we didn't do the experiments ourselves, it was in collaboration with the colleagues of Yoon. I think it's stable for at least 3 to 6, even 6 months. But I have to double check the exact timing of the shelf life.
And just very quickly, Anna asks, when would we expect to see the results of this trial?
We hope very soon, because actually currently we are doing a lot of analysis. So we hope by this summer that we will have all the data and that we can process all the results. So this trial is also high priority on our to-do list. So yes, we hope by the summer that we will have the results.
All right, I think we're out of time there, but I'm sure the ladies will be delighted to ask any questions later on. I believe we're taking a 15-minute break before Maria and Marco will introduce and partake in the Ask Us Anything session. Thank you very much. All the best.